How many of you know or knew someone with cancer? And I bet you some of those people were given ineffective treatments. About 1 million cancer patients each year are given ineffective treatments, emphasizing the importance of new and novel therapies. So, wouldn't it be awesome if our immune cells could sniff out the cancer? Well, turns out they can. Our immune system consists of many different cells with many different functions, all set to protect us. One cell in particular, called a macrophage, acts as the first line of defense. They identify and engulf harmful invaders, such as bacteria and cancer cells. I study a very specific macrophage with a very unique function. Through RNA sequencing, our lab has identified a specific macrophage that expresses the same receptor you and I use to smell the world around us. So my research focuses on identifying what this macrophage does and asks the question of how we can harness its power to increase cancer treatment efficacy. So the first thing I asked was if this special macrophage could affect tumor growth. So using mice that lack these macrophages, or sniffers, I gave them cancer and tracked their tumor growth. What I found was that these tumors without sniffers had significantly larger tumors when compared to normal mice. So, no sniffers, more cancer. So since macrophages are known to move towards invaders, I wanted to see if these macrophages could move towards cancer cells. So I put sniffing and non-sniffing macrophages together with cancer cells in a model system that mimics your body's environment. I found that only these sniffers were able to find the cancer cells, allowing us to conclude that these cancer cells might be producing a specific scent that these macrophages could find due to their nose-like feature. So once the macrophages get there, what do? I wanted to compare the immune cells inside of a tumor with sniffers to the immune cells inside of a tumor without sniffers. And what I found was that I did this by labeling each type of cell with a different color in order to identify the type of immune cells present in it. So as you can see, purple for macrophages and blue for other types of immune cells. I found that the tumors with sniffers had a larger amount of immune cells that were activated and ready to fight the cancer. So all this is nice and has been in mice so far, but unless we're trying to treat cancer in Mickey Mouse, we really needed to compare this to human patients. So using publicly available patient data, I found that patients with larger amounts of sniffers were able to respond better to immunotherapies and cancer treatments. So this leaves us with the question, if macrophages are able to be activated by smells, could we use odorants as a drug in order to increase our immune system and increase the response we get to cancer or treatments?